Good morning, everyone. Today, we will discuss a fundamental concept in genetics and molecular biology: mutation. A mutation is defined as a heritable change in the nucleotide sequence of an organism's genome. Mutations are the ultimate source of genetic diversity, but they can also cause diseases, including cancer and inherited disorders. In this presentation, we will classify mutations, explore their causes, and examine how cells respond to DNA damage. Let us begin with the primary categories of mutations. Mutations are broadly divided into two classes. First, point mutations are small changes affecting one or a few base pairs. Second, chromosomal rearrangements involve larger structural changes, such as deletions, duplications, inversions, or translocations of chromosome segments. Point mutations are more frequent, but chromosomal rearrangements often have more severe consequences because they can alter the expression or structure of many genes simultaneously. Both types are important in evolution and disease. Now, let us focus on point mutations. These include base substitution, where a single nucleotide is replaced by another. They also include small insertions and deletions, often called indels, which involve one to a few bases. A third type is the expansion of trinucleotide sequences, where a three-base repeat increases in number over generations, causing diseases such as Huntington's disease and Fragile X syndrome. Each subtype has unique molecular mechanisms and phenotypic effects. Base substitutions are classified by their consequences on the protein product. A silent mutation changes the codon but specifies the same amino acid due to the degeneracy of the genetic code. No effect on the protein. A missense mutation changes one amino acid to another, which may alter protein function depending on the location and chemical properties. A nonsense mutation creates a premature stop codon, leading to truncated, usually non-functional proteins. These distinctions are critical for understanding genetic diseases and their severity. Not all mutations occur in coding sequences. An RNA splicing mutation alters the recognition sites for splicing machinery, causing introns to be retained or exons skipped. This produces abnormal mRNA and often a non-functional protein. A promoter mutation changes the regulatory region where RNA polymerase binds. This can increase, decrease, or completely abolish gene transcription. Both types are common in inherited disorders and cancers, highlighting the importance of non-coding regions in gene regulation. Mutations are also classified by the cell type in which they occur. Germline mutations arise in eggs or sperm and are transmitted to offspring, causing inherited diseases. Somatic mutations occur in non-reproductive cells and are not passed to the next generation, but they accumulate with age and are responsible for most cancers. This distinction is fundamental in medical genetics. Germline mutations require family counseling, while somatic mutations guide oncology treatments like targeted therapy. Spontaneous mutations arise naturally due to errors during DNA replication, chemical instability of bases such as deamination or depurination, or oxidative damage from normal metabolism. Induced mutations are caused by external agents called mutagens, including radiation, chemicals, and viruses. Understanding this difference helps us assess genetic risk. For example, spontaneous mutation rates are relatively constant, but exposure to mutagens can dramatically increase mutation rates, leading to higher disease incidence. The two major causes of spontaneous mutations are: first, DNA replication errors. Despite proofreading by DNA polymerase, mistakes like base mismatches occur at a rate of about 1 in 10 to the 7th to 10 to the 8th nucleotides. Second, deficiency in the DNA repair system. If post-replication repair or mismatch repair fails, these errors become fixed as permanent mutations. Individuals with inherited repair deficiencies, such as xeroderma pigmentosum or Lynch syndrome, show dramatically increased cancer risk. Environmental mutagens are external agents that induce mutations. They include ionizing and non-ionizing radiation, chemical mutagens such as alkylating agents and base analogs, certain viruses that integrate into the genome and transposons, mobile genetic elements that can jump to new locations. Other factors like heat and oxidative stress also contribute. Reducing exposure to known mutagens, for instance, using sunscreen or avoiding tobacco, is a key public health strategy to prevent mutation-related diseases.
In a base substitution, we have two words like a transition and transversions. Transitions happen when a nucleotide of purine is replaced by another purine and or a nucleotide of pyrimidine replaced with another pyrimidine. And transversion happen when a nucleotide of purine replaced by pyrimidine or vice versa, a pyrimidine replaced by a purine. Let me introduce the five major categories of agents that cause induced mutation. First, rays or radiation. These include both ionizing radiation such as X-ray and gamma rays and non-ionizing radiation such as ultraviolet light. Both types damage DNA, but through different mechanisms. We will discuss them in detail in a text slide. Second, chemicals. There are thousands of chemical mutagens. Some are found in the environment, like benzopyrene in cigarette smoke. Others are industrial chemicals or chemotherapeutic drugs. Chemicals can cause mutation by alkylating DNA, intercalating between bases, or causing base oxidation. Third, viruses. Some viruses integrate into the host genome and can disrupt genes or activate oncogenes. For example, human papillomavirus or HPV can cause cervical cancer. Fourth, transposons. These are jumping genes, DNA sequences that can move from one location to another in a genome. When a transposon jumps into a gene, it can disrupt that gene's function. Fifth, other factors. These include things like heat, which can cause depurination and react oxygen species produced by normal metabolism. Understanding these categories helps us identify environmental and occupational hazards. It also helps us develop preventing strategies. Now, let's look more closely at radiation. Now let's discuss radiation in more detail. Radiation is divided into two main types, ionizing and non-ionizing. Ionizing radiation has enough energy to remove electrons from atoms and molecules, creating high reactive ions. This type of radiation includes X-rays, gamma rays, and alpha and beta particles from radioactive materials. Ionizing radiation damages DNA directly by breaking the sugar phosphate backbone, causing single strand or double strand breaks. It also damages DNA indirectly by creating reactive oxygen species that attack the bases. Double strand breaks are particularly dangerous because they can lead to chromosomal rearrangements, large deletions, or cell death. Sources of ionizing radiation include material X-rays, CT scans, nuclear accident, and natural backbone radiation from radon gas. Non-ionizing reaction and radiation has less energy. It cannot remove electrons, but it can still damage DNA. The most important example is ultraviolet light from the sun. UV light is absorbed by DNA and causes adjacent thymine bases to form covalent bands, creating thymine dimers. These dimers distort the DNA helix and block replication. In humans, thymine dimers are repaired by nucleotide excision repair. However, if repair fails, mutation can occur. Over time, UV-induced mutations can lead to skin cancer. As genetic counselors, we educate patients about protecting themselves from excessive UV exposure and about the risk of ionizing radiation from medical imaging.
Ultraviolet light is maximally absorbed by DNA at a wavelength of 254 nanometers. This energy causes covalent bonds to form between adjacent pyrimidine bases, thymine or cytosine, creating pyrimidine dimers. It also produces pyrimidine hydrates via water addition. These lesions distort the DNA helix and block replication. If not repaired, they cause characteristic C to T or CC to TT transitions. This is why cells have evolved dedicated repair systems like photolysis in some organisms and nucleotide excision repair in humans.
Now let's discuss chemical mutagens. There are thousands of chemicals that can damage DNA. They work through various mechanisms. Some chemicals are direct mutagens. They damage DNA directly. Others are indirect mutagens. They need to be metabolized by the body into reactive forms that can damage DNA. Chemical mutagens can cause a variety of DNA lesions. They can alkaline bases, meaning they add methyl or ethyl groups. They can oxidize bases, changing their structure. They can deaminate bases, converting cytosine to uracil, for example. They can also intercalate between DNA bases, causing insertion or deletions during replication. Many chemical mutagens are found in our environment. Tobacco smoke contains dozens of chemical mutagens, including benzopyrene. Processed food may contain nitrosamines. Certain industrial chemicals like benzene and formamide are non-mutagens and carcinogens. Some chemotherapy drugs such as cyclophosphamide and cisplatin are also mutagens. They kill cancer cells by damaging their DNA, but they can also cause secondary cancers years after. In the next slide, we will focus on a special mechanism called tautomeric shift. This is not caused by an external chemicals, but rather by the natural ability of DNA bases to exist in different chemical forms. Now let me explain an important concept, tautomeric shift. DNA bases can exist in two different chemical forms called tautomers. These forms are in equilibrium with each other. The common stable forms are called the keto form for thymine and guanine and the amino form for cytosine and adenine. In these standard forms, base pairing follows the rules. A pairs with Phe and G pairs with C. However, bases can temporarily shift to rare tautomeric forms. Thymine and guanine can shift from the keto form to the enol form. Cytosine and adenine can shift from the amino form to the imino form. These rare tautomers exist only for a very short time, but that is long enough to cause a mutation. How? When a base is in its rare tautomeric form, it can pair with the wrong partner. For example, thymine in an enol form can pair with guanine instead of adenine. Cytosine in an imino form can pair with adenine instead of guanine. If this happens during DNA replication, the DNA polymerase will insert the wrong complementary base. After the next round of replication, this error becomes a permanent mutation, a transition where a purine is replaced by a different purine or a pyrimidine by a different pyrimidine. Tautomeric shifts are a major source of spontaneous mutations. They occur naturally without any external mutagen. However, certain chemicals can increase the frequency of tautomeric shifts. Understanding tautomerism helps us understand the chemical basis of mutation. It is a beautiful example of how the chemistry of DNA directly affects genetics.
Chemical mutagens can act on DNA in two temporal contexts. Some mutagens only affect replicating DNA. They are incorporated or cause errors only when the replication fork passes. Others damage both replicating and non replicating DNA. They chemically modify bases regardless of replication status. This distinction is important in cancer chemotherapy. Agents that target rapidly dividing cells with replicating DNA are selectively toxic to tumors, while agents that damage non replicating DNA can cause mutations in quiescent cells, including stem cells.
In this slide, we look at how different mutagens affect two types of DNA, replicating DNA and non-replicating DNA. We have four classes of mutagens here: nitrous acid, acridine dyes, alkylating agents, and hydroxylating agents. The key point is that most of these agents are far more effective when DNA is actively replicating. First, nitrous acid. It deaminates bases, converting, for example, cytosine to uracil. On replicating DNA, this leads to permanent mutations after one round of replication. On non-replicating DNA, the damage sits there but remains silent until the cell divides. Second, acridine dyes, such as proflavin. These are intercalating agents. Their mutagenic effect is completely depends on replication. Why? Because insertion or deletions only occur when the replication machinery passes over the intercalated site. On non-replicating DNA, they cause almost no mutation. Third, alkylating agents like EMS. They add alkyl groups to bases. They do damage both replicating and non-replicating DNA, but the mutation rate is much higher during replication because error-prone repair or mispairing leads to fixed changes. On non-replicating DNA, lesions remain but are not yet converted into mutations. Fourth, hydroxylating agents like hydroxylamine. They especially modify cytosine. Again, the mispairing happens during replication, so the effect is strongly depends on DNA synthesis. In non-replicating DNA, the modified base exists but doesn't cause a heritable change until replication.
Nitrous acid is a deaminating agent. It removes amino groups from bases. Adenine becomes hypoxanthine, which pairs like guanine, causing an adenine to guanine transition. Cytosine becomes uracil, which pairs like thymine, causing a cytosine to thymine transition. Guanine becomes xanthine, which still pairs with cytosine and is usually less mutagenic. Deamination is also a frequent spontaneous event, but nitrous acid accelerates it dramatically. This chemical has been used historically in mutagenesis screens and illustrates how simple chemical modifications alter genetic information. Acridine dyes, such as proflavin and acridine orange, are flat, planar molecules that intercalate between DNA base pairs. This intercalation pushes the bases apart, lengthening and unwinding the helix. During replication, intercalation causes the DNA polymerase to slip, leading to insertions or deletions of one or more base pairs. Because indels often cause frameshift mutations, acridine dyes are extremely effective mutagens. They are used in genetics research to study reading frames and have also been used as antiseptics and fluorescent stains. Alkylating agents transfer alkyl groups, methyl or ethyl, to DNA bases. Examples include nitrogen mustard, sulfur mustard, methyl methane sulfonate, ethyl methane sulfonate, and nitrosoguanidine. These chemicals cause base mispairing, depurination, which is loss of bases, and strand breaks. Many are potent carcinogens and are found in industrial pollutants, cigarette smoke, and chemical weapons. Paradoxically, some alkylating agents are used in chemotherapy, such as cyclophosphamide, because they preferentially kill rapidly dividing cancer cells, though with significant side effects. Hydroxylating agents, such as hydroxylamine, add hydroxyl groups to bases. Hydroxylamine specifically reacts with cytosine to form hydroxylaminocytosine, which pairs like adenine rather than guanine. This causes GC to AT transition mutations with high specificity. Hydroxylamine does not significantly modify thymine, adenine, or guanine under typical conditions. Because of its specificity, hydroxylamine is a valuable tool in experimental mutagenesis for generating targeted mutations and studying structure-function relationships in proteins. To conclude this section, we have covered the major types and causes of mutations. We distinguish point mutations from chromosomal rearrangements. Within point mutations, we examine base substitutions, silent, missense, and nonsense, small indels, and trinucleotide expansions. We classified mutations by cell type, germline versus somatic, and by origin, spontaneous versus induced. We also reviewed physical mutagens such as radiation and chemical mutagens, including base analogs, deaminating agents, intercalators, alkylating agents, and hydroxylating compounds. Understanding these categories is essential for any graduate student in genetics, molecular biology, or medicine.